Awesome. Yeah, well, my name is Heidi Klumpe. I'm a postdoc at Boston University, and yeah, I'm excited to be here at this journal club talking about a recent paper that I worked on actually with JB. We're actually co-first authors. I just included the screenshot in case you want to look the paper up, and also to highlight that it was part of a virtual special issue on AI for synthetic biology. So if you're interested in those kinds of things, you could look this up. But basically to then provide some motivation for why we're interested in predicting single cell responses, I think as probably many of you are aware of, single cell variability can make it quite difficult to both understand. Sorry, I think there's some background noise. Oh, seems like we're good now. Okay, awesome. Single cell variability can make it quite difficult to both understand and control certain biological processes. So this is an example from Mary's group, where basically we might think of the input as the expression of this gene MARE that can then control the cell's response, how susceptible it is to antibiotic. And if you look at the population level, it's quite clear, okay, if we express MARE, the antibiotic susceptibility increases. But then there's this surprising effect that even when the system is sort of off, the levels of MARE are actually pulsing with time, which then means if you were to look at kind of across a population of cells treated with an antibiotic, what percentage of them die, you'd find that a fraction of them actually survive due to this pulsing. So even though the population is isogenic, these dynamic differences actually matter for predicting who will survive and who will die. And another example I think is really exciting is if we look at basically directed differentiation protocols, where again, maybe there's some input here, like a transcription factor ASCL1, which is controlling a neuronal fate. And despite sort of like understanding the biology and know that it's possible to reprogram with this transcription factor, these protocols are at best 20% efficient. So this is a paper from Stephen Quake's group where they did single cell RNA-seq to sort of understand why this happens. And again, as you might expect, if you look at the day 2 population, basically there are 2 colors. So they have done PCA on the whole transcriptome and found that even though the day 0 population seems to be quite similar to each other, there's actually 2 populations that emerge. So there's some heterogeneity that explains why they're not able to get sort of 100% reprogramming. And so I think when there's sort of something that's hard to understand in biology, usually the thing that helps us sort of control it and understand it better is to build some kind of model. And usually it's a mathematical model. But the thing that we're talking about here, single cell variability, is particularly hard to capture for at least a couple reasons. One is the kind of inherent stochasticity of the reactions and sort of fluctuations in the concentrations themselves, particularly in bacterial cells where the volumes are small. The sort of numbers of mRNA molecules or proteins can fluctuate quite a lot. And there's also this problem that many processes are sort of integrated across the cell. You may think that you have built a circuit or are studying a process that's like well isolated from other things that are going on, but actually a larger number of components potentially are involved, like the fact that ribosomes are shared sort of between all systems that are turning on proteins. And then there's other processes going on, which means that there are these unrelated dynamics that affect your process. Like the cell is cycling, it's also aging. So this could also affect sort of this cell's particular response to some input. And maybe there's some differences between these 2 major bullet points. The first one, the stochasticity, we sort of have no hope of predicting because it's truly random. But maybe these systematic patterns in the way that some processes affect each other or are sensitive to other dynamics are something we could learn, but maybe that would require that we survey or be able to directly measure a lot of things at the same time, or be able to write equations for a lot of types of interactions, and that would be quite challenging. So I think that's where machine learning is very exciting as an alternative to sort of learn this complex input-output relationship, basically what I add to the cell and then what it does as a response directly from the data without having to know all of these particular mechanistic details. So these are sort of wide-ranging examples that I think just highlight sort of the power and flexibility, as I'm sure everybody knows, but this is an example for earthquake detection from seismic activity data, where they're able to predict sort of where or not predict, I guess, post facto explain where they think the earthquake occurred. And it's this very particular pattern. Also, There was work to predict chemical properties from chemical structure. And something I think is really interesting about this example is the chemical structure is actually this string of characters, this like smiles notation, yet they're still able to predict certain properties with very reasonable accuracy. And of course, there's many examples from biology, but this is a cool recent one from Jim Collins' group called Bioautomated, where basically you can sort of predict or correlate biological sequences with any kind of property of interest. They look at translation efficiency and binding affinity for kind of just any set of biological sequences. So what this sort of suggests for kind of the question that I was interested in was it likely is possible to sort of do this prediction task that we're interested in, where basically we would have some sort of input, which is itself dynamic, sort of cartooned by it switching from black to gray. And then the cell has some response. And maybe we know its past response to this dynamic input, but then we would like to predict what is it going to do in the future. And I also wanted to point out that I guess maybe one problem with this is that we don't have the mechanistic detail. I mean, it's a benefit and that we don't have to directly model it, but does that somehow limit the usefulness of the prediction? And I think no, for at least two examples that I can think of. So one would be model predictive control, where you have a system that you want to control, and within your controller, you have a predictive model, such that you're able to optimize what's the best perturbation to sort of make my process match the set point or sort of follow the pattern that I want it to follow. Even without mechanistic information, you can still get sort of the best perturbation. And then also, particularly in mammalian cells, but I think it's quite common in biology, there are these like long signal transduction pathways connecting the outside of the cell to the inside. And even without kind of complete information about all of the complexes that form or the rates of phosphorylation, it would be quite cool if I just knew sort of these are the proteins that I need to add kind of in this particular dynamic or combinatorial fashion and get the cell response that I want. And what's sort of cool is I think that's how JB and I came together to be excited about this project. With his background in control theory, I think he was excited to build these controllers. And my PhD, I was studying developmental signaling pathways and trying very hard to model them. And it was just super exciting to think that maybe with some of the data that would not be hard to generate, we could actually get quality predictions. And so I think JB did a lot of the initial work to sort of identify what would be the appropriate machine learning method for doing this prediction task. He might have even, I think, presented on this in an earlier journal club, but basically showed with real cells that he could sort of record a cell's past history of responsiveness. Here, the input is light, and the output was the production of a fluorescent protein. And he was able to then predict how the cells would respond to a future light stimulus. But then at this point, because I was again thinking about these like long signal transduction pathways, I was curious, how would this architecture scale to more complex processes, or how would it work if we wanted to apply it to a different biological system? And so for the paper, we were sort of thinking about three questions, one of which is how do different types of noise affect the model predictions? So if we use this same architecture, does it perform, I guess, how far can we push it before it's no longer able to make a prediction? Also, how much information is required to make a good prediction, both in terms of what are the number of examples of responses the model needs to see to sort of learn how to make a prediction, but also how much access, how much information does it need about the cell's past responsiveness to make a good prediction of the future? Because again, for other applications, we might not be able to collect quite as much data or the same kinds of data. And then lastly, how would this scale to more complex circuits? So if there's quite a lot of steps between this input U and the output F, is it still able to learn the pattern of responsiveness and then make this prediction? So for, again, this paper, our particular approach was to simulate the data. Again, so we could sort of explore a lot of these different things. We were simulating this CCASR system, which is a common optogenetic system for turning on expression of a gene. You can use green light to induce the formation of this dimer, which turns on GFP if that's the way you engineer it. And then red light can turn it off. And again, this was nice because JB had shown that the model could work on this system. And then there was a great paper from Remy Shea and others where they'd fit a model with parameters. So we could produce biologically reasonable traces using this particular circuit architecture where light, basically when it is on, induces the formation of dimer with some rate. And then there's a Hill function which describes how the dimer turns on the fluorescent protein. And then in some of the cases, because we wanted to test sort of the ability of the model to sort of predict well, even in cases where there's varying cell responsiveness, we add this parameter E. And basically, it has dynamics that are independent of the light, but do affect how much of the reporter is expressed. And so just one more methods note. So basically what we're going to ask the model to, or this particular architecture to do kind of over and over again is first, we will give it some examples of responses. So we do the simulations where we have some known random light input, and then we simulate the responsiveness. We can divide this into a past and future. Then the model takes first as input the past responsiveness, so both the light and in this case sort of gene expression level. And then the encoder produces an encoded version of the cell past. And then we can show it the example of the future light. And then the decoder will predict what the future is. We used for most of the paper this same architecture where the encoder is a long short-term memory network, which is something that is well suited to working with dynamic data. And then for the decoder, we used a multi-layer perceptron. To train the model, we then compare its prediction to sort of our ground truth future, which is again just something we have from our simulation. So with the simulations, we generated 10,000 examples of single cell responses. You might see them referred to as cells in some of the figures. And so it's trained and tries to minimize data and explain those examples well. But then to test the quality of the prediction, we ask it to predict 10,000 examples that it has never seen before. Okay, so now with sort of this approach in mind, we looked at the first question, which is how well is the model able to infer response dynamics from noisy data? And the nice thing about using these simulations is we can explore the effects of stochasticity and different types of noise. So I'm showing the circuit here again, just as a reminder. And so even in the kind of deterministic case, again, we have red and green light, which is inducing the formation of the dimer. And then the reporter also responds with some delay because it needs some concentration of H to turn on. So even though these are quite smooth curves, sort of a non-trivial response maybe that the model needs to infer. We can then stochastically simulate these equations using the Gillespie algorithm. And again, it's similar. It's kind of clear that green turns it on and red turns it off, but there's more noise. And then lastly, we can add this additional parameter, the cell responsiveness, which is the green line, which you can see sort of not sensitive to light, but is going up and down and can change the response. Like in particular here, the fact that the responsiveness is high means that this dip in the amount of H doesn't really affect the responsiveness, which is actually quite different from the stochastic case where the responsiveness is constant. So ultimately what we did is we looked at kind of 4 different combinations of these things. And this plot is just showing how you can have kind of the same response input, but as you might expect, these different types of noise sort of affect the response. So the deterministic response is quite smooth. For measurement noise, we sort of, after the simulation, added this additive Gaussian, which again produces some variability. Then we can stochastically simulate and then add measurement noise. And then in the case that seems sort of probably most biological, we have the stochastic simulation of the equations, this varying cell responsiveness, and then the measurement noise. So how well does the model do when trained on these different types of data? Again, it can predict the deterministic responses nearly perfectly, which is, again, maybe not so surprising, but still pretty cool. And each of these subplots, we're showing kind of the past and the future time. The simulated response is the pink line, and then the predicted future is the black line. And you actually can't see the simulated future because the predicted future is exactly on top of it, because again, the error is close to 0. But when we add measurement noise, it becomes a little bit different. And first, I want to highlight that in any cases where there is like a little bit of randomness in how we simulate the future, there are actually multiple futures that are consistent with the same cell past. So to sort of get a reasonable estimate of the quality of the prediction, we wanted to ask how well does the model prediction compare to sort of kind of many possible futures. So we actually simulated the future of this test cell 1,000 times, and I'm plotting it with some transparency, which is sort of why there's this like thick cloud. So you can see like the additive Gaussian is just sort of like a sleeve around the average future. I'm also plotting the average as a gray line, which you can see clearly here. Because one way to think about this is that sort of the best the model can do, given that it's predicting a single value for the future, is to sort of capture the average. So that could be one way to sort of ask how well is it doing? Because obviously there's going to be some error here associated with the fact that it doesn't predict these like this random jitter, which of course it can't predict. But we see kind of here in the 95th percentile example that it's actually systematically sort of different than the average. And we think that this might mean that, again, the encoder is trying to make some sort of estimate of the cell's state of responsiveness, but also the current level of the fluorescent protein. So maybe it's made some sort of error in its estimate of sort of what the true state of the cell is. And so it's capturing the dynamics, but kind of with this error. So if we were to sort of summarize this, this is, again, sort of what the distribution of error, again, across these 1,000 examples in the test set would look like. Yeah, feel free to interrupt with questions if you have one. I think this is like choosing an error metric, I just wanted to say, is something that we thought a lot about. But we thought maybe the most intuitive thing was to use a normalized RMSE, where RMSE stands for the root mean squared error, which is basically a way of taking sort of the average squared error across all of those possible futures for particularly the measurement noise case. And then we've normalized it to what the value of the fluorescence actually was. So this is a bit like a percentage error. So what we see is that in the deterministic case, the median error is 0, because again, it predicted with really, really high accuracy. But what's cool is that the measurement noise then sort of sets some floor of error between, which it can't go below. Like the best it can do is sort of perfectly sort of get in the middle of that additive Gaussian noise. All right, so now we want to move to potentially the more interesting case when the underlying reactions are stochastic. So just for reference, these are the deterministic and the deterministic plus measurement noise cases. But then these are what the sort of the distribution of prediction quality looks like for the other cases. So we can look first here at the blue, where again, we have again, stochasticity in the equations. And I think one of the first things you'll see is that sort of this like cloud of possible futures is much broader because again, just sort of more unusual responses are likely. But if we look kind of at how well this black line sort of fits within this cloud or is able to predict the average, even in the median case, there's maybe some systematic errors at time 0, or it might even kind of become more wrong with time. This is particularly pronounced in the case where we also have this time-changing cell responsiveness. Again, there are examples where it can kind of do the best it can and predict the average nearly perfectly. But we see also here in this case, sort of in the 95th percentile, or one of the worst predicted examples from the test set, it's actually getting the dynamics kind of wrong, and the error is increasing with time. It can sort of correctly predict at the beginning, but not at the end. So this is then sort of a summary of sort of across the test set, what was the error for these 4 different cases. And again, as we sort of saw from the particular examples, the stochasticity increases the error considerably, and there's quite a long tail in terms of the quality. Somewhat surprisingly, and maybe not apparent just from looking at these single examples, is that the cell responsiveness, which I thought would be kind of like a really big problem, it's producing these dynamics again that are not controlled by the light. How is the model able to infer that? Actually didn't produce a huge decrease in quality relative to the circuit without. So it seems like the stochasticity of the equations themselves, or sorry, of the responses themselves is actually kind of the larger problem for prediction.
Heidi?
Yeah.
So at least in the example plots you showed, it seems like for a lot of these sort of switching of the induction of the light induction, it seems like it's often happening on a very like fast time scale relative to the dynamics. Is that something that sort of, I mean, did you guys, I mean, it could just be that if you do enough sampling, you see sort of the slower things, but I guess, did you guys notice a big effect of sort of how you design the set of inputs versus the quality of the predictions?
That's a great question. I think that's not actually something that we explored systematically. We sort of have this like particular way of generating a random light impulse, but we didn't. I think that's a great idea. It's something we're thinking about. It's like how do we could have, basically asked like we could make the dynamics of the light much longer or much shorter than the process itself, and sort of ask how it affects things.
Yeah,
because I guess obviously
for linear systems, that's like the main thing you do is you get a frequency response, and you see like as you change the frequency, how does the system's dynamics change? Like I wonder if you sort of gave it the linear tests of giving different frequencies, just like totally deterministic, would that work as well, better or worse than these sort of randomly generated inputs?
Yeah, I love that. I think that's a great question because yeah, with like light, it's like quite easy to kind of do these like random impulses where maybe you're like trying to get lots of different frequencies, but maybe if you're looking at a small molecule inducer, you've like, at best you can sort of pulse it up and down, and maybe it would be like the same across an entire microfluidic chip.
No, JB made the tunable inducer device for his PhD.
Yeah, I guess we could have done that. Awesome, thanks. Okay, any other questions? Okay, just the last thing I wanted to highlight that I thought was also sort of interesting is we can also look at the prediction error over time across the test set, which again, from the deterministic case, because it can predict the dynamics quite well, the error doesn't really increase with time. When we add the measurement noise, again, there's this sort of floor, which is just instituted by the fact that there's like randomness that the model can't be expected to predict. What's sort of interesting is that in the stochasticity cases, at time 0, it's able to do nearly as well as when there's measurement noise alone, which again makes sense. You know, a good prediction for the early time is like where you were before, so it has a reasonably good estimate. But this error increases with time. And this actually, I think, highlights maybe more of a difference of adding this changing in cell responsiveness, that sort of the envelope of error gets much larger. But in general, it's harder to predict the distant future than near future, which matches our intuition. Okay, and then for our, I guess the next question we were interested in is sort of what are the requirements for low error predictions? And maybe this is getting at a bit of some of what Noah was asking in terms of like, what kinds of data do you need to train the model well? And I think there are like so many interesting things we could do, but these are just, we were particularly interested in like training set size, and then how much of the past do you need to predict the future? So as you might expect, diversity in the training set is important for training sort of a general model. You want to give it lots of possible examples. And as a default, we were starting with 9,000 cells. But we saw that we could sort of reduce this down to even maybe 900 or 90 cells, where the distribution is here, and get reasonably good error. And it was only when we down to 1 cell that, again, we're looking at error that's on average 20% or so by average. But I think one of the reasons we can go down to so few examples is that we're simulating something that was true in the experimental case, which is that you can, on the device, measure the cell's responsiveness for a very long time, say 36 hours. But the model input is some like 3 hours of the past and then 2 hours of the future. So actually, for each training batch, we can sample a different 5-hour window out of that 36-hour response. So every single example contains many instances of the past and future, which is, again, maybe just kind of a strength of this approach. Maybe like you don't have to get much data in terms of number of cells, but just in terms of the time. But so then we were sort of curious, okay, what if we had like very, very short responses? So we could cut it in half. You only have 18 hours, or maybe you just have a 5-hour response. So every cell's response is indeed sort of a single example. And then we asked, okay, how does this scale with the number of examples? So here you can see that the 36-hour case, it does quite well as you reduce the number of examples. But if it truly is only 5 hours, you can't get away with only 10 cells or so. Oh, Ross put a question in the chat, presumably that only works for ergodic systems. Any other constraints? Sorry, Ross, do you want to elaborate or? Oh, maybe he's somewhere where he can't turn his microphone on, which is why he chatted it. Oh, okay. Okay. Please, yeah, bring it up in the Q&A. It sounds like an interesting question. Okay. But yeah, this again, these aren't necessarily like hard and fast numbers that like anyone would use for their system, but it was just interesting to see that this is sort of the limits in terms of what the model can be trained on. Something else we tested was using a validation set as a way to avoid presumably the overfitting that causes a problem when we're training on a very small number of examples. Basically, it can explain sort of this exotic and not representative set very, very well, which means it will not generalize well. So for the validation set, basically we had additional examples that we did not train on. So we train on, for example, in this case, 9,000 examples. And then there's some error sort of associated with those different models, but we only keep the model which minimizes error on the validation set, even though it's been trained on the training set. And sort of using this, again, additional type of held out data to predict the best model really means that you can reduce the number of examples, even for small or like short duration data, and still get reasonably good predictions. Okay. So beyond sort of thinking about sort of the number of examples or the duration, we were also curious, how much information about the past do you need to predict the future? Obviously, like the very, very distant past will not be useful for predicting the future, but the process itself might have some inherent timescale that it's important to sort of watch and understand before you're able to make a good prediction about the future. So we had originally been using examples of 3 hours of the past. We were able to reduce this down to 1 hour, and then good, again, reasonably good predictions. So that distant past was not as helpful, but when we reduced it to 30 or 15 minutes, again, then it basically sort of the prediction quality goes out the window. So again, there's some minimum amount of time it needs. Something we did not sort of systematically test here, but I think would be interesting to do next is, again, it's unclear if this is something about the inherent timescale of the process itself or the sampling frequency, because sort of inspired by JB's experimental setup, we simulated making observations every 5 minutes. So in the 15-minute case, it's 3 data points. In the 30-minute case, it's 6 data points. It's possible that the past 30 minutes of responsiveness would be enough information if we had sort of a higher frequency of data. And this will come up a little bit later, actually, in the next case, because then we test processes with sort of different inherent timescales. Okay, so for the final piece, we were interested in looking at how well this architecture could predict the dynamics of more complex circuits. And again, because I think those like long signal cascades are quite interesting, we thought, okay, we'll make sort of a simple cascade by adding this intermediate, which is also quite a common architecture in biology. So basically now this induced timer is turning on expression of I, which then turns on expression of F. There's also multiple parameter regimes. So we varied Ki, or sort of the sensitivity of F to I. And in the case where F is sort of very sensitive, it very quickly turns on because you don't need many molecules of I to induce F. And then it's slow to turn off because I needs to dilute before the fluorescent protein also stops being produced, versus a case where it's not very sensitive. So sort of a delay in turning on, but it decays quite quickly. So sort of skipping a lot of the work again. So we have these different architectures. We can do our Gillespie simulations, generate a bunch of data. We train models for the 2 cases. And then we look at the distribution of error in the test set. And we can compare sort of our 2 new circuits, so our cells that we think are behaving a little differently, to the simple activation case. And if we look here at the distribution of error, I think it's like largely within this sort of envelope where the blue is sort of here with a similar peak, and then the orange is maybe a little bit more overlapping with this tail that's still as good as those cases. We think some of the reason for sort of the systematic difference between these 2 cases is actually because of the distribution of fluorescence values in the test set. And again, because we're normalizing this sort of, the numbers that you're dividing by are either systematically small or large. And again, maybe this gets at kind of Noah's question about thinking about how to sort of structure the test and training sets. But basically, as you might expect, if there's a delay on and you have some set of like light impulses that you're using, it'll take longer to turn on. So on average, they were lower, versus if there was some delay in turning it off, the system tended to be on. But overall, we think this indicates that it performed kind of as well on the cascades as the simple activation. But this is sort of cool because then we repeated what we'd done in the previous case, because again, with these delays, there's now sort of a different inherent timescale of responsiveness in the system. So does that change the requirement for past information? And again, on the prior slide, we're looking at all of the cases where you have 3 hours of past information. And this dotted line is sort of showing, okay, the simple activation case with like complete information. And if we just look at the black dots, sorry, I think my legend is missing, but as we go down, we can reduce it to at least an hour and still get good quality predictions in the simple activation case. But in the case where there's a delay in the off, or sorry, the delay in how it's turned on, which is the orange. Even with as few as 1 hour, it's like not making a good prediction. And then in the blue case where there's a delay in, the delay off, I can do reasonably well with 1 hour, but it takes longer. So I guess the two cascades are different from each other, which is potentially interesting, but they both require more past information, which we might expect. Something we can't quite explain or we want to look more into is also kind of why this is non-monotonic. There's this like interesting sort of feature that sort of in the simple activation case, it explains early time points well and sort of late time points less well. But sort of in the limit of very little information about the past, it seems to then switch and then not explain early time points well, but explain late time points better, which I think maybe has something to do with this transition in terms of the quality of the error. Okay, and then as kind of the final challenge, we wanted to use this auto activation circuit, which implements positive feedback. And this is interesting. So we're not using the E variable here, which sort of introduces these changes in cell responsiveness because it kind of had like this explosion in difficulty of analysis. But this is already quite interesting. But the system has some hysteresis when you're turning it on, and then when you turn it off in terms of what's the steady state value of F. But basically what this means is that, say you turn the system on and then you turn it off, you can have bimodal outcomes in terms of how much it turns off. And again, this could be potentially a problem when you're kind of requiring your system to predict a single future. So already in sort of, we've seen many examples of this where there are unimodal outcomes and the single prediction is not able to predict the distribution, but is trying to make some estimate of the average. But this is particularly problematic when the outcomes are bimodal because now it's actually predicting sort of an impossible future because it's trying to interpolate between the two possible ones. So we had this idea of what if we sort of allowed a two-dimensional prediction where it could then maybe look at something like the distribution of future responses, or at least accommodate the fact that there's more than one possibility at a single time point. And so this second dimension would be sort of like multiple fluorescent values for each time point. And so to do this, we basically turned to a different decoder architecture because now we can think of our sort of predictions of the future as images because they're two-dimensional. So convolutional neural nets are sort of well suited to working with images. So we updated the architecture of the decoder. And then we also needed to sort of represent our ground truth futures also as images. So again, these traces now look like this. Basically, we apply some sort of Gaussian blur and do some binning to sort of get this square image that matches the shape of the output of the CNN. And what we were very happy to see is that this could improve the quality of the prediction. So I'll sort of step through this bit by bit, but this LSTM MLP is the decoder encoder that we used for the majority of the paper, which again can predict unimodal responses somewhat reasonably. But now if we train the CNN, it's making this prediction, which actually matches quite well the entire distribution of possible responses. So here, this is the image representation, again, from our ground truth simulations of what we expect the futures to be. And we can look at the distributions at this like 8-hour future, and they're like almost perfectly overlapping. But kind of more impressively, sort of in the bimodal case, where this architecture really does not work at all, the prediction matches the realizations, I think, in like a pretty stunning way. I think it's like kind of incredible that even sort of here, where it's like predicting that you get some sort of responses 2 or 3 hours in the future, it knows that at 4 or 5 hours, there's going to be some split. And that's where this like second outcome starts to emerge. And again, the overlapping at the final time point, the distributions are basically overlapping. So to summarize, these are the questions that we set out to answer and sort of what we found, which was that I guess obviously the different types of noise affect the model predictions, and that measurement noise sort of sets some sort of floor below which it's like hard to make a good prediction because again, it's inherently random. The stochasticity also sort of increases the error and maybe gives you this long tail, and potentially the stochasticity itself is more of a problem than the complexity of the network. And we also see that the stochasticity means that the prediction error increases with time. So it's really struggling to predict these sort of random dynamics into the future. And then in terms of how much information is required to make a good prediction, we saw that we could get down to quite a small number of cells, but there are particular trade-offs in terms of how long is a recording of an individual cell's response. And we could use the validation set to sort of protect us from overfitting in the limit of kind of smaller data set sizes. And also there is some minimum requirement of past information. We want to make a good quality prediction. And lastly, we can scale up to more complex circuits. So again, we saw that the cascade worked kind of nearly as well as the simple activation case, although it did require more past information, which makes sense because the kind of dynamics of the process are different. But I think overall, this like encoder decoder architecture is quite flexible. We could swap in a different type of decoder, and now we're able to make not only like a new prediction for a good type of circuit, but sort of an altogether different type of prediction where we're actually looking at the distribution of possible responses. And just to like give you a picture of like some things that we're thinking about going forward, I think it would be quite cool to think about this fate control problem in terms of like, I think there are a lot of things where there are sort of diverse inputs where sort of the combinations and the relative timing of those inputs matters. And we know that they're turning on, we think, some set of transcription factors via signaling pathway. And then there's a gene regulatory network that controls target genes. And then this produces phenotypic cell fates. And ideally, we just like to know, what do I need to kind of add to my cells in the dish to sort of make them behave in the way that I want? And then there are like lots of little prediction problems in here that maybe we can sort of work on to build up to this. And I think sort of this paper and also JSB's work show that like we can do a pretty good job maybe like predicting how transcription factor would produce a target gene, and not just in like a population average sense, but really at the single cell level. And so then we could think about, okay, can I predict how inputs then control amounts of transcription factors? In particular, can I modify this architecture to consider combinatorial inputs? It's already doing quite well with like timing, that what if I have like multiple things? And then lastly, can I think about how transcription factors control cell fates? Where again, maybe a cell fate is something phenotypic. And again, we weren't, our simulation data wasn't taking into account that maybe it was taken from a microscopy image, but you could imagine that you could look at the shape of the cell or something about its, I guess, geometry, or like a set of genes that are expressed, or maybe is it alive or dead, to predict its effect on cell fate. So yeah, thank you all for listening. I apologize again for being late, but I wanted to thank the people that could have made this possible, in particular JB, who you just give him coffee and then he will give you great ideas and bug-free code. Also Mary Dunlop, who was sort of the primary advisor we worked with on this, who really helped us put together the paper, and then also various people in the Dunlop lab who chatted and gave feedback. Yeah, thank you for your attention, and happy to take any questions.